Good afternoon, friends. So today we'll discuss about the evaluation of parental dosage form. The parental dosage forms are evaluated by different testing in sterility test, pyrogen test, clarity test, leakage test, assay, and stability test. So from that, today we'll discuss sterility test and pyrogen test. So the first one is sterility test. Before any evaluation parameter, before the study of any evaluation parameters, we have to consider the importance of that sterility study. So sterility study is used to determine the presence of microorganisms present in the formulations. So sterility testing. So this is the procedure for sterility testing. First one is random sampling. So after manufacturing of a dosage form, we have to select the random samples for testing. Then aseptic conditions we have to maintain to study the sterility test. Then growth of microorganisms is usually checked by incubating over a specified culture media at specified temperature for specified time. Then the principle of sterility test is if bacteria or fungi are placed in a medium and will provide the nutritive material and water and kept at a favorable temperature, the organism will grow and their presence can be checked by turbidity in a clear medium. Then steps involved in sterility testing. So for sterility testing, we have to carry this procedure. In that, the first one is selection of sample size. So first we have to select the samples from the batch, then the quantity of the samples for sterility testing, then method of testing. In that, two methods are used for the sterility testing. The first one is membrane filtration test and second one is direct inoculation test. Then last one is observations and results. So these are the four steps involved in sterility testing. So first one is selection of samples. So for after manufacturing of a parental dosage form, if the batch size is less than 100 containers, if the batch size is less than 100 containers, then we have to select 10% or for 4 containers, whichever is greater for sterility testing. If the batch size is 100 to 500 containers, then we have to select 10 containers for the sterility test. If batch size is greater than 500 containers, then we have to select 2% or 20 containers, whichever is less. For large volume parenterals, these are the some sampling values, and according to these values, we have to select the samples for sterility test. Then quantity per container, liquids. If the dosage form is less than 1 ml, if the dosage form is less than 1 ml, then we have to take whole the contents from each container for sterility testing. If 1 to 40 ml, the dosage form is 1 to 40 ml, then we have to take the half the contents of each container, but not less than 1 ml. Then 40 to 100 ml, 20 ml, greater than 100 ml, we have to take 10% of the contents, but not less than 20 ml. Then these are the some values, and these values are required to select the quantity of the sample for sterility testing. Then methods for sterility testing. In that, two methods are used for sterility testing. The first one is membrane filtration test and second one is direct inoculation test. In that membrane filtration test, we have to take the membrane filter for the sterility testing. In that, these are the steps. These steps are involved in the sterility test. For that, the first one is the filtration of test sample by passing aseptically through a membrane filter, and the membrane filter, the porosity of that membrane filter is 0.45 micron and the diameter is approximately 47 millimeter. So these specifications membrane filters we have to take for the sterility test, and through that membrane filter we have to filter the sample. After filtration, the membrane is removed aseptically from the metallic holder and divided into two equal halves. Then that filter paper membrane filter is divided into two equal halves, and the first half is transferred into culture media meant for fungi and incubated at 20 to 25 degrees Celsius for not less than 7 days, and the half part or half of the membrane filter is transferred into the fluid thioglycolate medium and incubated again for not less than 7 days at 30 to 35 degrees Celsius, and then check the growth of microorganism. So this is the composition or formula for fluid thioglycolate medium. L-cysteine, sodium chloride, dextrose monohydrate, agar, yeast extract, pancreatic digest of casing, sodium thioglycolate, resazurin sodium solution, and distilled water. So by using this formula, we can prepare the fluid thioglycolate medium for sterility test. Then second one is soybean casein digest medium. This is also used as a medium for the sterility test, and this is the formula for sodium casein digest medium. In that, pancreatic digest of casein, peptic digest of soybean meal, sodium chloride, dibasic potassium phosphate, dextrose, and distilled water is used. Then second one is direct inoculation method. In that direct inoculation method, we have to add the test sample directly into the culture medium. The specified quantity of sample is drawn aseptically from the container and then transferred into a vessel of culture medium. Then mix the liquid with medium and incubate for not less than 14 days. So that is the difference between membrane filtration and direct inoculation. In that membrane filtration test, we have to incubate for not less than 7 days, and in that direct inoculation test, we have to incubate the samples for not less than 14 days and then check or observe the growth of microorganism in medium. So after performing these two methods, membrane filtration test and direct inoculation test, we have to observe and then interpret the results. So first one is, if no evidence of microbial growth is found, then the product complies with the test for sterility. If there is evidence of microbial growth, then retesting is performed using the same number of samples, same volume to be tested and media as in the original test. If no evidence of microbial growth is found, then the product complies with the test for sterility. If again there is a evidence of microbial growth, so isolate and identify the microorganism. The second retest is performed using the twice the number of samples. In that first retest and first method, we have to take the same samples. In second retest, we have to take the double the samples, twice the number of samples. The preparation passes the sterility of test in case there is no evidence of microbial growth, and if there is a microbial growth, then we have to fail the test for sterility. So these are the observations and results. Then second one is Pyrogen test. So Pyrogen test is performed by using these three methods. The first one is sham test, second one is rabbit method, third one is lal test, that is limulus amoebocyte lysate test. So sham test is used for the selection of proper animals for the animal study, that is rabbit method, that is old method, and in vivo method. Third one is limulus amoebocyte lysate test, that is new method, and in vitro method. So first sham test is used to select the proper animals for the in vivo study. Then rabbit method, the rabbit method is old, and in vivo test. A pyrogen is defined as a fuel producing agent, that is the metabolic products of microorganisms. These pyrogens are soluble, filterable, thermostable, and non-volatile. The sources of pyrogens. The major sources of pyrogens are solvents, drugs, additives, apparatus used, then containers may be the sources of pyrogens, then method of storage in between preparation and sterilization may grow the pyrogens, then hence every item must be apyrogenic and method of storage must not allow any bacterial growth. So rabbit test consists of measuring the body temperature, rise in body temperature in healthy rabbits after the IV injection of sterile test solution. So in that rabbit method, we have to measure the body temperature of that rabbit. Then why rabbit is selected for the animal study or pyrogen study? Because other species are not predictable. These rabbits are economic, then similar pyrogenic response to humans and reproducible pyrogenic response. So these are the reasons, and due to that reasons we can select the rabbits for testing. Then requirements for rabbit pyrogen test. So rabbits should be healthy and mature. New Zealand or Belgian white rabbits are mostly used. Either sex can be used. Having the weight not less than 1.5 kg, must be individually housed between 20 to 23 degrees Celsius, not showing any body weight loss during the week of preceding the test. Equipments and materials used in test must be free from pyrogens. If these equipments and materials are not apyrogenic, then this should be made apyrogenic by heating at 250 degrees Celsius not less than 30 minutes, or any other suitable method to remove the pyrogens from equipments and materials. Then retaining boxes do not use any rabbits if a temperature is greater than 39.8 degrees Celsius. If the initial temperature is 39.8 degrees Celsius, then we can't use the rabbits for pyrogen test. The temperature variation greater than 0.2 degrees Celsius between two successive readings in the determination of initial temperature. So these are the requirements, and if the rabbit shows these two properties, then we can't select these rabbits for the testing. So this is the method. The whole method is carried out in air conditioned room. Dissolve the substance with pyrogen free saline solution, then warm the liquids up to 38.5 degrees Celsius. Then volume of injection is not less than 0.5 ml per kg or not more than 1 ml per kg of body weight. So this is the 0.5 ml to 1 ml per kg sample may be injected into the rabbits for study. So this is the procedure for rabbit study. In that, the clinical thermometer is inserted into the rectum of rabbit for recording the body temperature. Two normal readings of rectal temperature should be taken prior to the injection at an interval of half an hour, and its mean is calculated, which is the initial temperature of the rabbit. So to determine the initial temperature of rabbit, we have to determine two successive readings after half an hour. The sample is injected slowly through the ear vein of the rabbit at a volume of 0.5 to 1 ml per kg of body weight. Then record the temperature of each rabbit at an interval of 30 minutes, means for 3 hours, means we have to take the 6 readings for body temperature at every 30 minutes. The difference between the initial temperature and the maximum temperature shows the response. Then interpretation of results. After measuring the body temperature, if the sum of the responses of the group of 3 rabbits does not exceed 1.4 degrees Celsius, and if the response of individual rabbit is less than 0.6 degrees Celsius, then the preparation being examined passes the test. If the response of any rabbit is 0.6 degrees Celsius or more, or if the sum of the responses of 3 rabbits exceeds 1.4 degrees Celsius, continue the test using another 5 rabbits. If not more than 3 of the 8 rabbits show individual response of 0.6 degrees Celsius or more, and if the sum of the responses of group of 8 rabbits does not exceed 3.7 degrees Celsius, the preparation being examined passes the test. Then next one is the LAL test. LAL test is in vitro method. In vitro method means we have to perform this test on a lysate, Limulus amoebocyte lysis. Limulus genera of crab. Amoebocyte, this is the active component and it is derived from the crab blood cell. Then lysate, it is obtained by separating the amoebocyte from the plasma and then lysing them. It is in vitro method. It is used to detect the presence and concentration of pyrogens. So this is the quantitative method. So by using this LAL test, we can determine the amount of pyrogens. We can determine the quantity of pyrogens, which is not possible with the animal test. The lysate is collected by puncturing the hearts of mature crab. This method is simple, rapid and greater sensitivity than the rabbit test. So this is the procedure. In that, the preparation or test sample is added to the lysate. 0.1 ml test sample and 0.1 ml LAL agent, Limulus amoebocyte lysate reagent. Incubate for 1 hour at 37 degrees Celsius and mixture is analyzed. The results of the reaction is turbidity, precipitation or gel formation of mixture if the pyrogens are present. The rate of reaction is also depends on concentration of endotoxins, that is pyrogens, pH, temperature and presence of clotting enzymes and proteins in lysate. These are the advantages of in vitro test, that is LAL test. It is less variable. In vitro test easier to perform, more sensitive, less expensive, less time consuming and can give the quantitative results which is not possible with the in vivo study, that is rabbit study. So by using this LAL test, Limulus amoebocyte lysate test, we can determine the amount of pyrogens present into the formulation. Thank you.